RNA polymerase, which is responsible for transcription for the synthesis of RNA, it can initiate a new RNA chain on a DNA template and thus doesn't require a primer. In contrast of DNA polymerase, which require primer for the synthesis of new DNA. This requires that the DNA template be brought into the polymerase active site and held stably in a helical conformation. So the DNA must be in contact of the enzyme's active site, and that the initiating ribonucleotide be brought into the active site and held stably on the template, while the next nucleotide is presented with correct geometry for the polymerization to take place. So first it is prerequisite that the DNA, double helical DNA, must be brought to contact to the active site. Then the new ribonucleotide should be stably in contact with the polymerase. Then the second ribonucleotide, which will come to join the first ribonucleotide, that should also come in the correct geometry and correct orientation, and that too should be held stably in position so that the binding of these two ribonucleotides can take place. This is particularly difficult because RNA polymerase starts most transcripts with an A, means adenine, and that ribonucleotide binds the template nucleotide T with only 2 hydrogen bonds, because there are 2 hydrogen bonds between adenine and thymine. So in contrast to 3 hydrogen bonds between G and C. So here only 2 hydrogen bonds are present. So it is difficult to keep these in positions. Thus, the enzyme has to make specific interactions with the DNA template strand, the initiating ribonucleotide, and the second ribonucleotide to hold them all rigidly in the correct orientation to allow chemical attack by the incoming NTP. So all these things must be kept rigidly in space so that the joining of new nucleotide can take place. This is the reason that most transcripts start with the same nucleotide. During initial transcription, RNA polymerase produces and releases short RNA transcripts which are less than 10 nucleotides, and these transcripts are called this synthesis is called abortive synthesis before escaping the promoter. So when the polymerase will escape promoter, it will do the abortive synthesis, and then it enters the elongation phase and then synthesize the proper transcript. It has long been unclear how the enzyme's active site translocates along the DNA template during initial abortive cycles of transcription. 3 general models were proposed to explain this. The first model is the transient excursion model, and this transient excursion model proposes transient cycles of forward and reverse translocation of RNA polymerase. So this is transient cycles of forward, reverse, forward, reverse, forward, reverse. Thus, polymerase is thought to leave the promoter and translocates a short way along the DNA template, synthesizing a short transcript before aborting transcription, releasing the transcript, and returning to its original location on the promoter. So this will be repeated. So here in this you can see this is minus 35 element, and polymerase lies here. It starts synthesis here, and after synthesis it has moved forward, but after synthesis and release of this abortive DNA, it comes back and it comes at its original site, which is minus 35.